So, in this lecture, we'll learn that the cells of our body must communicate with one another in order to coordinate the functions of the organs, so the cells within the organs, and then also to coordinate organs between organ systems. Okay, so everything's functioning and doing that in a coordinated fashion. So if our organ systems are really functioning at the cellular level, it's cells that need to be able to communicate with one another. So they do that mainly through through chemical messengers. And so in this lecture, we're going to look at 4 types of chemical messengers, and then we'll also look at the receptor-mediated signal transduction. I'll explain that to you in a minute. And then we're also going to learn the difference between first and second messengers. Alrighty, so here we go. So when the cells of the body communicate with one another, some do it directly, okay, because they're physically bound to one another. So let me zoom this in on for you a little bit. So you can see the picture on the right shows gap junctions spanning across the membrane of both cells. And so it's really kind of producing a tunnel between one cell and another. So remember, a gap junction is a type of protein that has it's fluid filled in the middle. And so when you have a gap junction spanning the cell membranes of 2 adjacent cells, it's forming a tunnel between the two. So the cytoplasm is continuous between those 2 cells, and therefore materials can travel between the 2 cells. Okay, if you need a refresher on cell junctions, you want to go back to Canvas, and I put some of the videos from Khan Academy on the cell junctions. Okay, also surface markers, another type of protein, they can also link 2 cells together as well, and again, allowing communication between the 2. So that's a intercellular communication by direct association. So the cells are directly in contact with one another. Okay. But they also communicate indirectly, because I remember sometimes they communicate from distances apart from one another. Okay, and so it means what they're doing when they communicate indirectly, a chemical is traveling some distance from one cell to another, as you can see in this picture here. So chemical messengers, they're able to travel very short distances, so maybe just from one cell to the cell that's right next to it or adjacent to it, or they can end up traveling very long distances as well, and therefore they have to be carried by the blood in order to do that. So if you remember when we talked about positive feedback loop, the example I gave was a hormone being released from the brain, and then that was traveling through the blood to the uterus, and the target cells in there, the hormone would bind to the smooth muscle cells of the uterus, causing contractions. So that's an actual type of chemical messenger binding to its specific target receptors. So it's just traveling through the blood indiscriminately, but it happens to affect the cells that have a receptor on the cell membrane, where then the effect will take place. So that's how a chemical kind of, quote, knows where to go. It doesn't really know. Again, the target cell, the cell that's going to accept that chemical messenger, has a receptor on the cell membrane. So we call that the target cell. Okay, so a chemical messenger will bind with any cell that has its specific receptor on it. Okay, so remember the term target cell. So first, let's look at 4 types of chemical messengers, and then we'll look into the receptors and the response that gets produced inside of the cell. So the first type of chemical messenger I'll talk about is a paracrine messenger. So a paracrine messenger travels a short distance. So it's just a chemical messenger that one cell makes. It secretes, that means it leaves through the cell membrane. It could be secreted through exocytosis. and it gets into the interstitial fluid, and the target cell is very close. Looks like I cut off the L in the word cell there on local target cell. Sorry about that. But this is then how tissues can start to act as a unit. So if the secreting cell is producing a chemical that it's affecting all the local target cells around it, then the response being created happens pretty much at the same time with those cells. Okay, so it can really help tissue cells to respond almost as a unit. So remember that the messenger is some type of chemical, and this is what we're looking at, the types of chemicals that they can become. Another term we might use is ligand. Ligand is just a general term for a molecule that binds to a receptor. Okay, so you might see sometimes in the book the chemical messenger being referred to as a ligand. Okay, the second type is a neurotransmitter. So neurotransmitters are also short-range signaling molecules, and you already learned about these pretty extensively when we studied synapses. We saw that a presynaptic cell will make a neurotransmitter, and it gets secreted by exocytosis into the synaptic cleft, and that was just the space between the presynaptic cell and the postsynaptic cell. The postsynaptic cell has the receptor, right? So here, short-range targeting cell, it's just traveling across the synaptic cleft, and that's a very, very small space. The next one are hormones. Hormones are long-range messengers. Okay, so usually the secreting cell is from the endocrine system. So we would call that an endocrine cell. So an endocrine organ, an endocrine gland would secrete it. It could be the adrenal gland, it could be the pancreas, it could be, there's lots of different types of endocrine cells, the pituitary, something like that, right? But their target cells might be a long distance away. And so remember that target cells have those receptors that are specific to the hormone, just like the paracrines and the neurotransmitters. So that's how, again, the target organ with the target cells, that's how the hormone kind of quote unquote knows where to go. It's actually just binding to the receptor on the surface of the cell of that distant organ. Okay, again, the same example with the oxytocin. The oxytocin is a hormone being secreted from the pituitary gland, and so that's an endocrine organ traveling through the blood, and the distant target cell are the smooth muscle cells in the uterus. So the fourth one is kind of a combination of the second and the third one, a neurohormone. Neurohormones are also long-range messengers. They behave very much like hormones, and they are a hormone, but it's a hormone that was made inside of a neuron. So neurons make neurotransmitters, but neurotransmitters only go into the synaptic cleft. A neurohormone is a hormone made by a neuron, but that messenger molecule gets secreted into the blood. Right, so we just call it a neurotransmitter when it only goes into the synaptic cleft. We call it a hormone when it travels in the blood. But to distinguish it from the other hormones we talked about, again, we're kind of referring to where it was made. So a neurohormone is made by neurons, and it secretes it into the blood. A regular hormone is made by an endocrine cell, and I see my picture there still says endocrine cell. I should change that. So this is made by a neuron that gets secreted into the blood. An endocrine cell is from epithelial tissue. So other hormones usually are mostly made in epithelial tissue. All right. So once a signaling molecule attaches to the receptor, then a response has to occur inside of the cell. Okay, So remember, the other thing to always remember is that these chemical messengers never enter the cell. Okay, these are what we call hydrophilic messengers. So that means they're water soluble, water loving, and they cannot cross through the lipid bilayer of the cell membrane. And so therefore they don't enter. Later in the semester, we will actually see some chemical messengers that can pass through a membrane. But for now, this is the most common, so we want to start with the hydrophilic chemical messengers. So again, the signal from the chemical messenger, or what we might call a ligand, the signal gets conveyed into the cell's interior. The messenger itself does not. It stays on the outside. So by that chemical messenger binding to the receptor on the outside, we refer to that as the first messenger. Okay, and that's going to bring about a cellular response inside of the cell because the message gets transduced or across the cell membrane inside the cell. And how does it do that? 3 main ways that we're going to go over in general. One, the opening and closing of gated channels. That should sound familiar to you. Another one might be that binding of the receptor activates an enzyme inside of the cell. Okay, and then another one is by initiating that binding of this receptor initiates a cascade of protein production inside of the cell. So again, we'll look at each one of these a little more closely. Okay, so this one, let's take a close-up view of this. This one you've seen already. This is the one where the first messenger binds to the receptor and opens the gated channel. So we went over this really well with neurotransmitters. So it's the same thing, but a hormone can do this, a paracrine can do this. It just depends on what area of the body that we're talking about. So in the picture there, you can see the green gated channel. That's a chemically gated channel. We saw that on the outside is the receptor. So the extracellular messenger is also the first messenger. So that's your neurotransmitter, your hormone, your paracrine, whatever it is. Opens a channel, ions enter, and the desired result occurs inside of the cell. Just remember though, maybe this first messenger and receptor complex, maybe what it does is it closes a channel, preventing ions from entering. Maybe it opens a channel and ions leave, right? As in the case of potassium. So lots of different things can happen here. And again, that really just depends on the specificity of the type of first messenger and its receptor. Okay, so again, you're not seeing that green ball, the first messenger, enter the cell. You're seeing ions enter the cell. I keep repeating it, so that might be an important point. Okay, the first messengers in this case do not enter the cell. All right. In this example, the first messenger, again, is one of the chemicals we've been talking about. It could be a paracrine, it can be a hormone. But the receptor protein is an enzyme of the inside of the cell membrane. So unless the first messenger is bound on the receptor side in the extracellular fluid, the enzyme remains inactive. But once it binds, the enzyme becomes activated. And what that means is, remember, enzymes are protein. And so the binding of this first messenger causes a chemical response with the protein, and it changes its shape. So the atoms within the protein kind of reform bonds, and it just kind of changes their shape along the inside of the cell membrane. Okay. And so in this case, the enzyme is a protein kinase. So you're going to hear this throughout the semester. A protein kinase is a type of enzyme that phosphorylates other enzymes. That means it adds a phosphate to them. So the enzyme, by adding a phosphate to them, ends up activating a protein inside of the cell. Right? This activated protein, you can see the red one there in the picture, becomes the second messenger. And it's the second messenger now that is causing a series of events, of chemical reactions, that's bringing about the desired response inside of the cell. So that's what we mean by signal transduction. The first messenger is bringing the signal to the cell, causing in order to produce a response inside of the cell. But since the first messenger can't enter it, we've got this series of events here, number 2 and number 3 in the photo, that's spreading the message from the outside of the cell to the inside of the cell. So it's doing that by activating enzymes and things. And I just want to point out again, we called the enzyme a protein kinase, ASE. So again, always remember in biology, when you see a word with ASE on the end, it's usually an enzyme. Okay, here's a specific example of this type of receptor enzyme. So this pathway is called a tyrosine kinase pathway. And so again, it's specific. This is an actual type of receptor enzyme complex, a tyrosine kinase receptor enzyme. So you can see on the extracellular side, there's actually a double receptor. We had seen this a little earlier as well. The receptor side requires the binding of 2 first messengers, or 2 signaling molecules. If only one binds, then it's not going to activate the inside of the cell. This is one way that our body uses to just kind of control how quickly a response occurs. And so you can imagine that if there's not enough of the signaling molecule produced, whether it's the hormone or the paracrine, whatever it is, then you're not going to get a response. So what we want to do is wait until we have enough of this hormone produced, so it kind of slows down the response time a little bit. So they bind, and that means what's happening then on the inside of the cell is that this enzyme called tyrosine kinase, it self-phosphorylates. You can see it takes some phosphate from an ATP, and that activates a protein kinase active site, which takes an inactive protein, one of the red ones on the left side there, phosphorylates that, and activates that protein. And that active protein now is the second messenger causing some type of cellular response. Right? This is what happens with insulin. Insulin is a hormone that would bind to a receptor on the outside, to a tyrosine kinase receptor enzyme. That would then activate the tyrosine kinase on the inside of the cell, and a second messenger would be activated, causing channels to open to allow glucose in. Okay, so a little bit of a familiar response. I think most of you know about insulin. This is exactly the way that insulin affects the inside of a cell. Okay, a receptor enzyme tyrosine kinase pathway. One thing to be really clear on when I go through these examples is where is the first messenger and where is the second messenger. Okay, and if I give you specifics of what a second messenger is, like I am in the next one, you have to remember what that name of that molecule is, and then why it's a second messenger. Okay, here's the third type that I want to go over. This is a G protein coupled receptor, or a GPCR. Okay, a GPCR gets much more complex, but it produces a big protein response inside of the cell. Okay, so here you can see in the picture, in the blue protein up on the left side through the cell membrane. On the outside in the extracellular fluid is the receptor side. Okay, and you can see our first messenger is binding to it. Okay, it can be a hormone or something like that. The actual protein does span through the cell membrane, and it kind of squiggles itself through the inside. It actually passes through the cell membrane 7 times. If you count how many times that passes through, you'll see 7. And then it ends on the inside of the cell membrane, on the intracellular side, with a subunit protein complex. Okay, so what that means is there's a complex of 3 protein subunits, and it's designated there. One is an alpha, one is a beta, one is a gamma. And those subunits then are going to pass the signal through the inside of the membrane to an effector protein. Okay. Once the effector protein gets activated, it produces the second messenger. That's the intracellular messenger. The first messenger is the extracellular messenger, causing this complex to occur, this movement of the subunit from the G protein, causing that effector protein to produce the second messenger. Okay. In this figure, you can see the little red bulb indicating the second messenger. In reality, that looks like it's just producing one, but in reality, the effector protein can produce many second messengers. And then each of those second messengers will activate a protein kinase. Okay, and again, that enzyme will phosphorylate other molecules. So each one of those is doing that, activating other protein molecules, which is producing the desired cellular response, producing the molecules that we want inside of there. So again, this kind of steps you through in general what a protein coupled receptor does. And let me give you a more specific example. I'm going to actually label in here the second messenger this time, and we'll even label the effector protein. So again, look at the top. We have a hydrophilic hormone binding to the receptor. Okay, they're specific, so even the shape of the hormone, it has the complementary shape of the receptor. Okay, so again, that means they're specific. I called it a hydrophilic hormone because it is soluble in water. And remember, the middle of the plasma membrane is non-polar. That means it's hydrophobic. So the hormone could not pass through on its own. So it's the first messenger. It binds to the outside. Okay. What it's going to do then, once it binds, that causes the G protein complex to dissociate. And in this case, it's the alpha subunit that dissociates. In a different cell with a different receptor complex protein or with a different hormone receptor combination, it might dissociate the gamma. It might dissociate the beta, or both the beta and the gamma. So again, a different response gets produced with the different specific receptor types. So in this case, an alpha moved, and that activated the effector protein, the green, just on the inside of the cell membrane. That's called adenylyl cyclase. Another enzyme. Adenylyl cyclase. What adenylyl cyclase does is it converts a molecule of ATP into what's called cyclic AMP. And it does that by taking two phosphates off of the ATP, kicking them off, and what's left is a 1-phosphate associated with the adenosine molecule. So now that's called adenosine monophosphate, AMP. So let's pause here a minute. Go over to the left side where you see my little chemical equation there. This is a little bit of simple math. Okay, ATP, if you remove 2 phosphate groups, which you see on the right side of the arrow, we remove 2, that leaves us with AMP, adenosine monophosphate. Adenosine triphosphate is 3 ATPs, tri is 3, and we result with AMP. We took 2 off of the 3, and now we get 1. Mono means 1. Cyclic AMP. Okay, our cyclic AMP is the second messenger. So the cyclic AMP now stimulates a series of pre-programmed chemical steps that bring about the response dictated by that first messenger in the beginning. Okay, and so these proteins might be important in a metabolic pathway. Okay, so again, I just want to say it again. The first messenger is what is stimulating this response, but it's not able to cross the plasma membrane. It's hydrophilic, so it's soluble in water. It can't pass through the nonpolar middle portion of the cell membrane. So it just passes its signal indirectly through the G protein complex in this case. Okay, the alpha dissociates, activating the adenylyl cyclase, our effector protein. Adenylyl cyclase converts ATP into cyclic AMP. Cyclic AMP now is the second messenger. Remember that. Okay, and cyclic AMP now can take inactivated protein kinases and activate them, which in turn activates other proteins, okay, which is producing your cellular response. We're making some kind of protein inside of this cell here. All righty. I had mentioned previously that, again, this is a very simple picture of what occurs. It looks very linear. One protein activates a cyclic AMP, which activates another protein kinase. But in reality, it's more of a cascade of events of one protein activating several cyclic AMP, which can activate several more proteins. And that's illustrated pretty well here in this figure. Okay, all of these figures are in your book. So I just want to explain a little further about this cascading effect. Okay, what it's doing, a cascading effect, is producing amplification of the response. If you amplify something, you increase the amount of it, right? And so the point here is by just taking one chemical messenger, one first messenger on the outside, and stimulating that G protein, that can actually stimulate several adenylyl cyclases, which can then, each one of those can activate cyclic AMP and activate several cyclic AMP. Each one of those cyclic AMPs can activate several protein kinases. Each one of those can phosphorylate another protein. And each one of those can activate another protein. So you can see at the top there, our one first messenger ended up amplifying the response. Each different protein, each different molecule on the inside was able to amplify the response. So starting with one chemical messenger, we ended up with millions of product at the end. I have 10 million at the end there. That's what I mean by amplification. Make sure that you can explain what amplification is and how something like a G protein complex can amplify the response. Okay, so in reality, on the outside of our cell, there will be several receptors. So each one of those are amplifying the response inside of the cell. Okay, so that's it for this lecture in Chapter 4. There's a lot more. When you read the book in Chapter 4, there's a lot more information there, and it gives more specifics on paracrines and hormones and other messenger pathways. It also compares the nervous system to the endocrine system. We're not going to worry about that stuff right now. So you're not responsible for those items right now in this unit. Later on, we will look at all of those things, but in context with the organ system when we study that organ system. So I think it's going to make it a little bit easier for you to understand those specifics when we look at the actual organ system and discuss it in context. Okay, so right now, just the material that we went over in this lecture, you can pair it with what's written in the book if you need a little bit more information, and that's all you're responsible for in the chapter. Same with all of the information. If it's on Canvas, if it's in a video, that's the information that I want you to know. And by the way, if you're having a little trouble understanding the G protein complex here, the G protein couple receptors, I did put on Canvas one of those Khan Academy videos where they go in a little bit more detail. There's a section in the Khan Academy where the video zooms in and it looks like he is talking about something in the video that you can't see. Just be patient. It's going to zoom back out. It's about 30 seconds of that. It's going to zoom back out and you're going to be able to see it. Okay? So thank you, everybody.